Hello, Miss Dawn. I don't have your voice.
Good afternoon.
How you doing?
I'm good. I'm kind of tired today, though, for some reason. So
Oh,
lots of running around this week. So I guess that's it.
yeah. Miss Dawn, I want to talk to you about the QC. Is it okay to ask you some question about the QC class?
Sure. Well, let's wait till the end of class.
Okay, sure. Hey, Aisha. You doing okay?
Yeah, I'm good. How are you?
Fine. How is your grandma?
She passed away.
Oh, really? I'm so sorry, Tisha.
Okay, thank you.
I'm so sorry to hear that, hon. I didn't know that.
I know we haven't seen each other in a while.
I'm so sorry.
It's okay. Thank you though.
Oh my goodness. I know it was hard for you. It takes time, Tisha.
A long time.
Yeah, I just went home this weekend. I flew down this weekend. Saw the fam.
Oh, that's sad.
Yeah, I'm getting through it. School's a
I
good
know.
distraction. School's a good distraction.
That's
Yeah.
good. Yeah.
I just decided that it's okay to cry. After my dad died, there were some days that just out of the blue, I'd just cry. I mean, it's like you're just sad. I mean, what do you say?
Yeah. Mhm.
It's okay to be sad,
Definitely
You
happened.
love them.
Yeah.
But it's hard.
Mm.
Hey, Don,
Yes.
I got a question about our article.
Okay.
Is there a specific length that you want? Because I have a feeling mine's going to be kind of lengthy.
The article summary, there's no length. It's going to depend on the length of your article. If you pick a 10-page article, that summary will be longer than the summary probably from a 5-page article. It just depends on what you picked.
Mine's got like a lot of like cellular processes in it, and it's like I'm having a hard time kind of like simplifying that. So,
Well, maybe you need to look at another article. I mean,
I like that article.
oh, okay. Well, I mean, that's the challenge of the rad bio literature is sometimes it is very deep and it's hard to understand some of the things they're, trying to tell you. Sometimes you just have to go to another reference to help you understand it.
Yeah.
So that's not unusual. That's not unusual to have to do that. All right, who are we missing? Shamarva and somebody. Markavia? Oh, I'm admitting her, so there you go. All right, so we're gonna go ahead and get started. If all y'all would turn on your video so I can see you. I hope everybody had a good Labor Day weekend.
Yeah.
Did
How about
something
you, Donna?
fun. At least the weather was cool. The weather was cool and the humidity was low, so that was good. It was a nice weekend. So hopefully you got some rest or did something fun. Wanted to remind you of a couple of things. First, that your article selection is due by end of day today. Many of you have already submitted. Make sure you post it on the discussion forum for your classmates to see and into the assignments. I think that everybody that submitted an article, it's all been approved, but you just want to make sure that these last few students who are looking don't pull your article. So go ahead and post it to the discussion forum if you haven't already.
It's for Rad Bio, right? I'm a little kind of between classes, so it's Rad Bio, not quality control, right?
Not quality control, Rad, but we're
Or to post the article to.
yeah, post your article that you selected for summary to the discussion forum, just so everybody else can see it and won't pick it. Somebody else won't pick it. So that's due tonight. Now, today we're going to review cell biology, cell structure, mitosis, meiosis, and all of this should be review. I know you've had all this material some other place at some other time. So our first test is this weekend. So it's due on Saturday. So it will be available on Thursday evening. It'll be available like at 4 o'clock on Thursday. And then it will be available till end of day Saturday. So what we're going to do to start with, I'm going to share my screen and we're going to play Kahoot. So sign in to Kahoot.
You're my favorite because we played Kahoot.
We're reviewing. We're going to review. We're going to review from last time. Let me see if I can do this. All right, I need one more player who's not in. Now I'm down to 10. All right, so here we go. Who am I missing? Nobody's going to tell me.
Not me.
Huh?
It's not me. I'm here.
I'm in.
Everybody think they're in? The number keeps varying. The highest we've had is 11. There should be 12 of you in here. And now it's back down to 10 again. So people keep getting booted out.
Well, if you let your
Well,
phone go idle, it will.
shouldn't it be 13 of us?
Well, yeah, but everybody's not in class today because there's only 13 people on this Zoom with me. So that means there's 12 of y'all.
No, you're not counted, Don.
Huh?
You're not counted.
Oh, okay, whatever. All right, well, we're starting. So here we go.
Let's do it.
All right, hydrogen, the most abundant atom.
Hey Don, I think it's me. It says I'm in, but it's not letting me on it.
All right, keep trying.
Mm.
Smallest subdivision of a compound is a molecule. Smallest subdivision of an element is an atom. The parenchymal tissue is the active tissue of the organ. That's what does the work of the organ. Okay, connective tissue is most abundant. All right, water. Remember, we're 85% water. It's organic, it contains carbon. Water does not contain carbon. Catabolism, breaking down macromolecules. Anabolism is building up. Okay, so proteins are long chain amino acids. There are only 22 amino acids in the body. All right, good. All right, good. DNA is only in the nucleus, but RNA is in both the nucleus and the cytoplasm. All right, let's see who did well. All right, great.
Congratulations to you guys.
So take a screenshot of your score. There's an assignment that says classwork 2 and upload it there when class is over. So good job. So now we're going to start on can y'all see this?
Yep.
Yes.
All right. So now we're going to start on the unit that is reviewing cell biology. And like I said, this is a review. I know you've had all this information before. Remember when we talked about the field of radiation biology? Radiation biology consists of lots of different scientists. Cell biologists who can investigate the function of the cell and how radiation affects the cell. And then we have physicists who describe the radiation and the energy of the radiation, the type of the radiation. So it's important that we review cell structure so that you have a good understanding of it. So that's what we're going to do today. So when we look at the components of a cell, we're going to talk about the cell membrane, the nucleus, the cytoplasm, and all of the cytoplasm, well, not all, but most of the cytoplasmic organelles, or at least the major cytoplasmic organelles. So that's what we're going to be covering. And we're going to start with the cell membrane. And the cell membrane is sometimes called the plasma membrane. And the cell membrane is actually what keeps the cell intact. It keeps all of the organelles, the nucleus, everything inside the cell. And it actually is a barrier that separates the intracellular and the extracellular environment. So one way of thinking of it is just the bag that holds all the components of the cell. It's the cellular encasement. It primarily consists of lipids and proteins. That's why we have fats throughout our body, because all the cell membranes are made of lipids. And in terms of its function, it does function as a barricade. It keeps things that don't need to come into the cell out of the cell. It also controls the passage of materials into the cell, either via active or passive transport. And if you remember when we talked about drugs entering cells in pharmacology, some drugs will go by passive transport, which is basically diffusion, but active transport requires a protein to move the molecule across that cell membrane. So again, cell membrane just houses all of the structures of the cell.
Can you repeat what you said for passive? You kind of broke up.
Oh, I'm sorry. If it's passive transport, it just moves because of concentration, okay? So it's like a diffusion through the cell membrane. But if it's active transport, it has to have energy. So it usually has a carrier protein that will move it through the cell membrane, but it requires energy. Passive transport does not require the cell to use any energy.
It'd be like sodium, potassium pump kind of thing.
Yeah, they would probably go by passive instead of active transport, I would think. But I'm not 100% certain. I would have to look that up. Okay, the nucleus. So here we have a diagram of the cell, and we see this large sort of circular structure in the cell, and that is the nucleus. Now, the nucleus we know is the control center of the cell because that's where the DNA is. The nucleus is separated from the cytoplasm by the nuclear envelope. So it has its own encasement that keeps it separate from the cytoplasm, except when cell division occurs. When cell division occurs, the nuclear envelope breaks down. Now, I've already said that in the nucleus, we have DNA. DNA is our genetic information. The DNA makes up the genes that make up the chromosomes. So all of our genetic information is housed with the DNA in the cell nucleus. It also has inside the nucleus a structure attached to the nuclear membrane that's called the nucleolus. And the nucleolus is a rounded structure. I'm not sure. Oh, here it is, sort of illustrated here. We've got the entire nucleus. We've got the nuclear membrane, the nuclear pores. Then we have the nucleolus. But the nucleolus contains RNA. What kind of RNA is in the nucleus?
mRNA.
mRNA. So the messenger RNA is in the nucleolus, and which is within the nucleus. So all of those things are the job of the nucleus. When we typically think of it, we probably most often focus on the DNA, but the RNA also is in the nucleus because remember, it has to carry out the instructions of the DNA. And also the nucleus forms the ribosomes. When we think of ribosomes, what do? We'll get to them later, but anybody know what the ribosomes do?
They're like carriers.
They communicate.
Yeah, they go between the cytoplasm and the nucleus.
But what happened? What do the ribosomes do?
They transport.
No.
They help digest thingies.
Nope.
Synthesize
Assimil-
protein.
Protein
metabolism.
synthesis. When you think
Oh,
about the
protein.
when you think about the ribosomes, the thing that should immediately come to mind is protein synthesis. Okay, so next we're going to look generally at the cytoplasm. Now, the cytoplasm is all of the protoplasm outside the cell nucleus, but within the cell membrane. So if the nucleus contains the brains, the cytoplasm contains everything else, all the workers. So in the cytoplasm, lots of things happen. This is where metabolism takes place. So cell metabolism occurs in the cytoplasm. That's where new materials are assembled, protein synthesis, carbohydrate synthesis. That's where materials are broken down to produce energy. Remember catabolism, where we break down macromolecules. What are the three end products of catabolism? One we know is energy. May I remember the other two?
Is that the aerobic and anaerobic?
No, I'm just talking, what I'm asking is what are the three end products of catabolism? So when a macromolecule is broken down, you end up with three things. One is energy. This
Water and
is water
carbon dioxide.
and carbon dioxide. So energy, water, carbon dioxide. Those are the three end products of catabolism. In the cytoplasm, also things are packaged to distribute. Things are shipped outside the cell membrane. And of course, you have to have a cleanup crew. So one of the functions of all the organelles in the cytoplasm is to eliminate waste products. Now, there has to be a cleanup crew.
Lysosome.
The lysosomes, that's right. So the cytoplasm really is sort of gel-like. It's mainly water, but it does have other things. It has nutrients for the cell. It has waste products. It does have electrolytes. And I guess if we looked generally at the job of the cytoplasm, it holds the organelles. That's where all the different cytoplasmic organelles are. And again, they are the workers. So since they are the workers, that's where metabolism takes place. And we've already introduced the term anabolism and catabolism, where we're either building up or breaking down macromolecules. So we'll start to look now at some of the cytoplasmic organelles, and we're not going to cover all of them, but we'll cover the main ones. And the first one that we look at is the endoplasmic reticulum. And the endoplasmic reticulum can either be rough or smooth. So what would differentiate? How would you know looking at a diagram of the cell? Because we see on this cellular diagram, and it's a little small, but we see this is the smooth endoplasmic reticulum. And then we see that this is the rough endoplasmic reticulum.
Ribosomes?
I'm sorry, what'd you say?
The ribosomes?
The ribosomes. So which contains the ribosomes?
The rough?
Rough.
Rough. Because we see these little dot-like structures on the exterior of the rough endoplasmic reticulum, and that is the ribosomes. So we associate the rough endoplasmic reticulum with protein synthesis because that's the job of the ribosomes. The smooth endoplasmic reticulum does not have the ribosomes, and we associate it with lipid synthesis. So fats are constructed in the smooth endoplasmic reticulum. So when we think about the endoplasmic reticulum, it is a series of channels. And so things are built and things are transported. But again, the rough endoplasmic reticulum has the ribosome, so we associate it with protein synthesis, the smooth with lipid synthesis. So the next we have is the Golgi apparatus. And in this particular diagram here, it's showing, we're looking again at the endoplasmic reticulum just to show you the difference. Here's the rough with the ribosomes. Here's the nucleus. And here we see the Golgi apparatus. So the function of the Golgi apparatus is carbohydrate and glycoprotein synthesis. So it's also building things. It's also channel of communication because we can export things from the cell via the Golgi apparatus. And remember, the endoplasmic reticulum is also a channel of communication, and the Golgi apparatus is connected to the endoplasmic reticulum. In terms of appearance, it looks like flattened sacs. So 4 to 6 flattened sacs, sort of stacked on top of each other. And it also forms the lysosomes. So the next of the cytoplasmic organelles are the mitochondria. And the mitochondria, these are the ones that everybody always remembers because you remember the term, the powerhouse of the cell. So here in this illustration, we have 2 mitochondria shown. They're relatively big structures. They are described as bean-shaped or peanut-shaped, but their function is to supply energy to the cell. Next, we have the lysosomes, and the lysosomes are smaller structures. They're sort of rounded, kind of described as pea-like structures. I think Chad already told us they are the garbage men or the garbage disposals in the cell. So they remove the waste products of the cell. They digest those cellular waste products, cellular fragments, cellular garbage. So they contain enzymes that will break down the waste products. All right, we've already mentioned the ribosomes. The ribosomes are these tiny little dot-like structures that are attached to the rough endoplasmic reticulum. We know that their job is protein synthesis. The messenger RNA brings information to the ribosomes, and the transfer RNA then accumulates the required amino acids to build the proteins. The next of the organelles that we have are the centrosomes. And the centrosomes are associated here with the centrioles. They are located near the nucleus. So do any of you remember what the centrioles do in the cell? What's their job?
Don, I sent you a private message. Have you seen it yet?
I have not. So does anybody remember what the centrosomes and the centrioles do?
They initiate cell division.
They don't initiate cell division, but they do have a part in cell division. They form the spindle fibers during cell division. So we'll talk a little bit more about that in just a few minutes. Now, if you look, you'll see there are other structures in the cell. We see microtubules. Microtubules are sometimes described as.
Oh wait, hey Don, that made me
Yes.
think when you said that. Don't the centrioles got something to do with the microtubules or something like that? Don't the centrioles help form the microtubules or something, or play a part with them?
You know, I'm not sure.
I think it does. I don't know. I'm not. I
That
mean, I
didn't
don't
come
know.
to
I just wouldn't.
mind. Look,
I could be wrong. I don't know.
oh, listen, I could be wrong too. I'll just be honest. Hang on, let me look in the textbook a second and see if it gives any indication of that. So let's see, the centrioles.
Isn't that in mitosis?
Yeah, their job occurs during mitosis, but I was just trying to see if says the centrosome made of two centrioles and the microtubules. So yes, you're right. The microtubules are a part of the centrosome. So good. I didn't remember that. So anyway, there are other microtubules in the cell. If you see, and here's one illustrated. So the microtubules, basically, they also provide pathways for products to move within the cell, and they support the cellular shape. Let's see what else is in here that we're just mentioned briefly. Cilia, what does cilia do?
For motion, or for
Movement. They can aid in movement of the cell because they're on the outside. They're on the outside of the cell membrane. And so they can assist movement. See, is there anything else in here that we have? The peroxisome. Anybody know what they do? They break down toxic materials. So if anything toxic gets into the cell, they'll break it down. So the cell doesn't die from poison, I guess. Let's see. I think that we've covered pretty much everything. But again, all this is review. You've had all of this before. So hopefully you're remembering it.
Forever ago, Dawn.
Forever ago. Oh no. Well, it should be familiar at least. So that's good. All right. So we've covered some of the functions of the different organelles. Now we're going to look at cell division. And cell division can either be mitosis or meiosis depending on cell type. Mitosis is cell division of somatic cells. So what does that mean, somatic cells? What is a somatic cell?
It relates to the body.
Cell division.
It's just cells of the body, the general body cells, the normal body cells. And another way of looking at it is that the somatic cells in your body are all the cells that aren't the reproductive cells. So if the reproductive cells are dividing, it's called meiosis. Does anybody know another term for the reproductive cells? What's another thing, a name that they could be called?
Germal site?
They could be called the germ cells. So yes. So somatic cells are cells of the body. Reproductive cells can also be called germ cells, or they can also just be called the sex cells. I mean, it just depends. But you typically reproductive or germ cells would be the terms that you would see. So we're going to look at mitosis. So we're going to start with somatic cell division. Look at the 4 stages of cell division and what happens in each stage. So with the process of cell division, the 4 stages are prophase, metaphase, anaphase, and telophase. And we see them diagrammed out here. However, cells aren't dividing all of the time. The period between cell divisions is called interphase. So when cells are dividing, they're in mitosis and they would be in one of those 4 stages. But when they're not dividing, they are in the interphase period or the intermitotic period between divisions. Okay? How often do cells divide?
All the time.
All cells divide all the time.
Every 10 seconds.
Nope. Anybody else got any guesses?
Every day.
Well, cells are dividing every day, but how about this? What is the lifespan
Red blood cells lifespan like that.
of a white blood cell? How long do your white blood cells last?
Is it 16 days?
Something like 3 to 5 days. So your white blood cells are dividing every 3 to 5 days. So somebody mentioned red blood cells. What's the lifespan of your red blood cell?
Like 120, man.
120 days. Your red blood cells aren't dividing every 120 days. What's the lifespan of your nerve cells?
2 to 3 days.
Forever. They don't divide.
I say they like
Nerve cells you're born with, it's what you have unless you kill them some way. So when
like
you're born, you've got all the nerve cells you're ever going to have.
like with burns too. When people burn out, and they never get that feeling again. Something like that.
I can't hear you, Kiara. You're breaking up. So say it again.
So like when people get burns, you know, it damages the burns, first, second, or third, and those cells never go back sometimes.
Yes, 3rd-degree burns that kill all the skin cells in that area, they won't come back because you've killed all the cells that would ultimately replace. Skin cells last about every 3 weeks. So you get a new set of skin every 3 weeks. So the answer to the question of when do cells divide, the answer is it depends on the cell type. Some cells, particularly epithelial cells that are coverings and linings, divide relatively frequently. The blood cells, the white blood cells specifically, divide very frequently. Some cells only divide as needed. Muscle cells only divide as needed. Nerve cells don't divide. So again, how often do cells divide? The answer is it depends. And if you want to know the mitotic period for a particular cell type, you'd have to look it up. I don't have any idea of what they all are, but they're different. Okay, some types of cells divide much more rapidly than others. But when the somatic cells are dividing, they go again through these 4 stages: prophase, metaphase, anaphase, telophase. So we're going to look at those stages and what happens. All right, so prophase. Prophase is the first stage of cell division. It is the longest stage of cell division. And this is where the chromosomes first become visible. Normally, if you were looking at cells under a microscope, you would not be able to see the chromosomes. The chromosomes are only visible during mitosis. So in prophase, the individual chromosomes become visible. The centriole pairs migrate to opposite ends of the cell. because you're going to get ready. So here we see within the nucleus, the chromosomes are visible. The centriole pairs, one have gone to one side, the others have gone to the other side. The nucleolus that is within the nucleus disappears, so you can't see it, and the nuclear envelope breaks down. So now there is no barrier between the nucleus and the cytoplasm. So all of this occurs in prophase. Then we have metaphase, so the second phase of cell division. The chromosomes are most visible during metaphase. So for scientists who are studying the chromosomes, they would try to halt the cell division in metaphase so they could best see them. In metaphase, remember the nuclear envelopes broke down, we can see the chromosomes, but in metaphase, the chromosomes all line up across the center of the cell or the equator of the cell. The centromere on the chromosomes duplicate. What is the centromere? What does the centromere on the chromosome do? Anybody know?
Doesn't it split them apart, like pull them apart?
It's part of the pulling them apart. But if we look at this illustration of a chromosome, the ends of the chromosome are called the arms of the chromosome. The arms of the chromosome are held together at the center by the centromere.
It lines them up.
It doesn't really line them up, it's just part of them. Now, in cell division, these chromosomes are ultimately going to break apart. So for each chromosome to have its own centromere, the centromere has to duplicate. So we have twice the genetic material because before cell division can occur, you have to have duplicate chromosomes, but we need duplicate centromeres so that the chromosomes can be pulled to the opposite end of the cell. So the centromeres duplicate, and the spindle fibers that are produced attach to each centromere. So you have the centriole pairs at each side. Remember, they move to each side of the cell during the first stage of cell division. They now have produced spindle fibers that have attached to each centromere, because now each chromosome has 2 centromeres. So,
Hey, Don.
yes. What's your question?
I just want you said that prophase was the longest phase, correct?
Yes, prophase is the longest phase of cell division. Metaphase,
Okay.
the second phase, is where the chromosomes are best seen. Okay. All right, so when this happens, we are now ending metaphase, going to anaphase. So during anaphase, the chromosomes split apart, with one chromosome going toward one centriole pair, and the other chromosome going toward the other. So the chromosomes have split apart. Now they're moving along the spindle fibers toward the opposite end of the cell. So during anaphase, things are just moving. The chromosomes are moving. They're trying to get to each side of the cell. Then comes telophase. Now, telophase is the last phase of cell division. During telophase, the chromosomes get to each side. So half the chromosomes get to one side, the other half the chromosomes get to the other side. We don't need the spindle fibers to pull them anymore. They've made it to where they need to go. So the spindle fibers go away. At that point, the centrioles duplicate and the nuclear envelope reforms around the chromosomes. The chromosomes then become invisible. So they diffuse back into the nucleus so we can't see them anymore. And the last stage of telophase, or the last thing that happens in telophase, is cytokinesis. And cytokinesis simply means the cytoplasm splits and moves apart. So we now have two separate cells. So lots of things happen when we are wrapping up cell division. You got to get the chromosomes where they're going, get the nuclear envelope surrounding them again, duplicating the centrioles, and then the cell breaking apart. The end result of mitosis is two daughter cells identical to the parent cell. The cell that starts to divide is called the parent cell. At the end of mitosis, you have two daughter cells that are exactly like it. So again, we start with one cell, we end up with two cells that were identical to the one that we started with.
Don, hey Don,
Yes.
I like to always remember that by the owl eyes. It looks like an owl's eye.
Oh. Well, that would never have occurred to me, but if it helps you remember it, great. All right, so then when the cell is finished dividing, we then have interphase. So remember, most of the lifetime of the cell is not spent in cell division. How long do you think cell division takes? Because it's pretty consistent across all cell types.
24 hours.
24 hours. So Kiara says 24 hours. Anybody else got a guess?
64 hours.
64 hours. Well, y'all are way too long. Cell division only takes about an hour. So in the lifetime of a cell, only a very small portion is spent in mitosis. The rest of the cell's life is spent in interphase. So during interphase, we have cell growth, but the most important thing that happens during interphase is that the DNA replicates, because the cell has to reproduce or double the chromosomes before it can go into cell division. So when the DNA replicates, and think about the DNA molecule. The DNA molecule is a macromolecule. It is huge. Thousands and thousands and thousands of nucleotides make up a single DNA molecule. But during interphase, when the DNA has to reproduce the chromosomes, it literally, remember it's a double helix, it has to split and it has to unwind, and then the molecule is split. Then the required base partners are picked up, and then the sugar phosphate chain is rebuilt on each of the two daughter DNA helixes. So it's quite complex. So like I said, the DNA, here's the standard DNA molecule. It's going to uncoil or unwind. The hydrogen bonds between the rungs, or not between the rungs, yeah, between the base molecules breaks apart. It separates. So here we see the original side rails are separated. There are enzymes that cause the new complementary base pairs to be built, and they are brought to the molecule, paired in the proper order, and the nucleotides are rebuilt. So when you have the new strands of the DNA, half of the strand is old, it's from the original. The other half of the strand is new, it's been built. So you get two identical strands of DNA. Now, how long do you think that would?
I give up.
You give up. Well, it only took an hour for cell division, but it takes about 15 hours to rebuild or to replicate the DNA. So takes a little while, but again, that's a relatively short amount of time when you consider the lifespan of some functional cells. They may not divide for days or weeks or months. So in interphase, part of the time is built with cell growth, part of the time is built or is used with DNA replication, but it really doesn't take that long. 15 hours is not too long considering.
Well, considering that they're not getting paid for it.
They're just working away and not getting paid for anything. All right. Let's take a break. We'll take a 10-minute break. I have it's 2:26, so 2:36 we'll be back and we will continue on. Okay. So hopefully everybody is back and ready to get going again.
Yeah, let's knock this out.
Knock this out. There we go. All right. So we're talking about cell division, and there are two types of cell division, mitosis and meiosis. Mitosis is somatic cell division. Meiosis is reproductive cell division. Mitosis ends up with two daughter cells identical to the parent cell. Meiosis is a reductive cell division in which we end up with 4 cells with only half of the genetic information of the original parent cell. So with meiosis, basically what happens is that the cell undergoes 2 consecutive cell divisions, but there is no replication of the DNA in the middle. So if we start here, Here we have a cell, the DNA has replicated. The first cell division occurs, giving us 2 daughter cells that are identical to the parent. Then, however, the cell undergoes a division again, and since there's no DNA replication, you end up with 4 daughter cells, and they're sometimes referred to as the granddaughter cells, with only half of the genetic information.
Which are the diploids, right?
You're going from the diploid number to the haploid number. So you have half of the genetic information. Siobhan, you had a question, but when it wouldn't let me click on the "you've raised your hand" bit. So what do you need? Or do you
Am I the only one that is hearing you really slowly and kind of choppy?
So I don't,
No,
so is
I heard it too.
connection
I had
not
really
good?
And what's being said? No, ma'am. I've went out. I've come in. I don't know what else to do. But if it's other people, I just wanted to know because I
It
can't.
may just be the internet connection here. It's
Hey
not
Don, go outside and climb up on one of them trees in the woods out there and see if you can fix it.
really.
I climb up. Dang, Marcus.
I need to get my disc and start climbing the trees. That's probably the truth. No, it's not giving me any indication that my signal was bad. So I'm sorry if you're having trouble hearing me.
I feel like it only happens from time to time.
Oh,
Like,
okay.
right now you sound completely normal.
Okay. That's what I'm saying,
Yeah.
because usually if the connection's bad, it will tell me. But right now, I mean, it hadn't told me. So
No, you go for
if we have
fine, and then like you just drop off.
okay, sorry. All right, so anyway, for meiosis, with the reproductive cells, we have 2 consecutive mitotic divisions, basically. If they're still going through those 4 stages, prophase, metaphase, anaphase, telophase. The first time we get 2 cells with a full set of chromosomes, but because the DNA does not replicate, after the second division, we end up with 4 granddaughter cells with half the genetic information. So the end result of meiosis, like we said, 4 granddaughter cells, half the genetic information, because in conception, these cells will join with cells from the other parent to create a full set of chromosomes.
Can you say that again? So, because what now?
When conception occurs, when the egg and the sperm join, both the egg and the sperm only have what we would call the haploid number, or half the normal chromosomes. But when conception occurs, they join together, and the child that's conceived would have a full set of chromosomes. Half the chromosomes from the father, half the chromosomes from the mother. One thing that's important to note about the granddaughter cells is that they are not identical. You end up with 4 granddaughter cells, but they are not identical because crossing over occurs, and that is when genes change places on what they call sister chromosomes. This is basically why if you have siblings, you're not all identical. You got half of your chromosomes from your father, you got half of your chromosomes from your mother, but because those 4 granddaughter cells are not identical, we're all a little different. We may have some characteristics that are similar, but we have many characteristics that are different, and that's because of that crossing over.
So is that where brothers and sisters hate each other?
What about twins?
This is not a psychology class. I have no idea.
So when they have
I love them.
twins, what about twins, Don? Like when they have twins, so that means everything's identical, correct, within the cell?
If they are identical twins, they have a germ cell that literally, after conception, the germ cell splits. So they have exactly the same set of chromosomes.
Right. Okay.
Fraternal twins, obviously, that's not true. But for identical twins, yes, the egg splits. So, so their chromosomes would be exactly the same.
Oh, cool.
One thing that's also kind of interesting is with the female germ cells, the egg, after these divisions occur, you have 4 cells, but really only one of them is viable. The other 3 are considered non-functional. So I think that's kind of interesting. But with the spermatogonia, they're all functional. But for the eggs, only one of the 4 is functional. Okay, so any questions about meiosis and mitosis? I mean, you just have to remember the stages, remember the steps.
Yeah, I got one right quick. Could you go back? Could you say specify which one is the haploids and the diploids again?
We'll come to this later when we look at the unit on genetics. But when we talk about the diploid number simply means having a full set of chromosomes. Well, a full set of chromosomes is 23 pairs or 46 chromosomes. Okay?
Right.
So somatic cells have the haploid number, not the haploid, excuse me, the diploid number of chromosomes. 23 pairs, 46 total. 22 somatic chromosomes, 1 set of sex chromosomes, or 22 pairs of autosomes or somatic chromosomes, 1 pair of sex chromosomes. The reproductive cells have the haploid number, or half that, meaning they have 23 chromosomes, not 23 pairs. So does that make sense?
Yep.
Okay. All right. So the last thing we're going to look at is what's called the cell cycle. And honestly, I'm not sure if y'all have covered the cell cycle in any other classes. Is this a term that that you're familiar with?
Yeah.
Okay.
Yeah.
Okay, good. So this is review as well. All right. So when we talk about the cell cycle, basically what we're doing is describing all the different stages in a cell's lifespan. And how you look at the cell cycle depends on what your profession is, I guess. So geneticists look at the cell cycle as only 2 stages. They look at mitosis as 1 stage and interphase as the 2nd stage. So basically, the cell is dividing, or in all this other time, it's not. That's just the way they look at it. We know what happens in interphase. We know that's when the DNA replicates. But for a geneticist, that's the way they look at it. Cells dividing, it's in mitosis, or it's not, it's in interphase. The cell biologist breaks down interphase. So the cell biologist looks at the cell cycle in terms of 4 stages. The first is mitosis, which is that prophase, metaphase, anaphase, telophase, so the cell's dividing. But then the cell biologist breaks interphase down into 3 separate stages, G1, S, and G2. So M phase we know. M phase is mitosis. Prophase, metaphase, anaphase, telophase. That's when the cell is dividing. That takes approximately 1 hour out of the cell's lifetime. So all of the other time in the cell's life is spent in the intermitotic period, interphase, which the cell biologist breaks down into G1, S, and G2. Now, depending on the reference, G can stand for either growth or gap. So the phase G1 is very, very variable in length. It can be as little as a few hours up to many years. So the length of G1 is variable. And the main thing that is happening in G1 is RNA synthesis. So G1 is the time period after mitosis, before S phase, which is DNA replication. And again, the length of G1 is widely variable depending on cell type. When a cell leaves G1 phase, it goes into S phase. Now, S phase is the time period of primarily DNA replication. So we've already talked about how DNA replicates. The molecule unwinds, it breaks apart, new bases are picked up and attached, and then you have 2 DNA molecules that are identical. So S phase takes about 15 hours. When S phase is completed, the cell biologist would say that the cell moves into G2. And G2 is, again, second gap phase or the second growth phase. G2 is somewhere between 1 and 5 hours. And the main things that are happening in G2 are protein synthesis and RNA synthesis again. Once the cell leaves G2, it goes into mitosis. So the cell would divide. So if we look at the time frame, mitosis takes about an hour. S phase takes 15 hours. G2 at its longest takes 5 hours. So we can account for 21 hours in the lifetime of a cell. So for white blood cells, if we say white blood cells have a lifespan of 3 days, how many hours are in 3 days?
72.
72.
72, all right. So there's 72 hours and we can account for 21 of them. So that means there's 51 hours unaccounted for. So for 51 hours, basically that white blood cell is in G1, okay? Synthesizing RNA.
Or G2, right? Or
I'm sorry?
G2 as well, right? G1 or G2, right?
No, G1, because G2 we accounted for. We said if we assume that G2 was 5 full hours. So G2 at 5 hours, S phase at 15, M phase at 1.
Okay.
4 hours.
All right. Yeah.
Okay, well, what about nerve cells? We said nerve cells,
They just roll.
they're stuck.
They
They don't
never
ever
die. They just
divide.
rolling.
They forever.
They're forever and ever stuck. So they're for cells like that who have a very long lifespan and they either don't divide or they very seldom divide. There's a stage called G sub 0. And G sub 0 replaces G1 in cell populations that are considered non-dividing or very slowly dividing. Because let's face it, however old you are, all your nerve cells have basically been stuck in G1 that entire time period. So I'm 61 years old. All my nerve cells are stuck in G1 for 61 years. So they would technically call that G sub 0. They're not really going to progress through the different stages in the cell cycle.
Hey, Dawn.
Yes.
So like when people come in for like x-rays, they'll say, oh, my knee been hurting all these years or whatever. That's because the nerves was damaged years ago and no progress has occurred.
Well,
that's what that is.
Well, there's no really way to say that because it might not be nerves. It could be vascular. It could be skeletal. So you wouldn't really know what the source of their pain is. I mean, if you've killed off nerve cells, basically they wouldn't have feeling or they would have tingling maybe. With that kind of instance, it would be hard to pinpoint a problem just based on what they've said. But if, the nerve cells don't regenerate. So think about patients at the Shepherd Spinal Center. They've been in horrific car accidents and their spinal cord is damaged. Well, then they're paralyzed. Those cells have died and they're not regenerating. I know there's been a lot of research on trying to make nerve cells regenerate. I do not know how much success they've had because they don't divide. So if they can't divide, you can't replenish the population. That's why spinal injuries are basically permanent, in a lot of cases because they won't divide. But anyway, these are the stages of the cell cycle. How often a cell divides depends on the specific cell type. A couple of things to note. If you are irradiating a patient, cells in the body are at different stages of cell division or different stages in the cell cycle. So the most radiosensitive stage in the lifetime of any cell is M phase, when it is in mitosis. So when the cell is dividing, it's most easily damaged by radiation. I'm sorry?
I was gonna say like in pregnancy, that first trimester, that would be like that too, right?
Well, we'll get to that in one of the later units, but yes, the first trimester of a pregnancy is the most radiosensitive time frame of the pregnancy. But that's because cells are rapidly dividing. So a lot of cells are in mitosis. Yes. So M phase, the most radiosensitive phase of the cell cycle. The most radioresistant phase, or when cells would be least damaged, is late S. Late S phase is when the DNA has just finished replicating. It's getting ready to go into G2. So, most sensitive M phase, most resistant late S. So again, M, G1, S, G2. For cells that don't divide, G1 is replaced with G sub 0, which basically just means they're stuck there for a long time. So, some things to remember. When we think about the cell, the majority of the cell is cytoplasm, and the majority of the cytoplasm is water. Cells have to reproduce. It's essential to cell survival. If cells cannot reproduce, then their cell line will die. The cell is most vulnerable to radiation during reproduction, which is M phase. And since the chromosomes are most visible during metaphase, when they're all lined up across the equator of the cell, radiation-induced damage to the chromosomes would be best analyzed during that stage. Okay. So any questions?
I got one more question, Don. When we were talking about the cytoplasm, the three things were water, energy, and hydrogen, correct? Or was it like the out products?
Okay, no, the end products of catabolism, okay, which is the breaking down of macromolecules, was water, energy, and carbon dioxide.
Gotcha. Thank you.
So when those macromolecules are broken down, you end up with those three things. You end up with the energy for cell metabolism, you end up with water, and you end up with carbon dioxide, which is a waste product.
Gotcha. Thank you.
Okay. All right, so any other questions? All right, I'm going to share my screen. We're going to do, if it will.
Oh, and Don, your Dropbox isn't up for the screenshot. It's not a Dropbox for it.
There's not? Oh, well, I'll correct that as soon as we finish. I thought I had set it up so it would allow it. Okay, here's a cell diagram. So let's go through. Oh, and before we look at this, I want to tell you all the best, your best reference for this is for this material is chapter 6 in the radiation protection book. So definitely go through chapter 6, and because it's got all the information about cell structures, the different chemical composition of the body, and all of that. So make sure you're reading chapter 6. So let's look at these, and I realize some of it didn't copy very well. So what is number 1? Anybody know?
Mitochondria.
Mitochondria.
Yeah.
All right, so number 1 is the mitochondria. So what's the purpose of the mitochondria?
Powerhouse.
Powerhouse, energy production. All right. Number 2, we see there's a
Centrioles.
Intrameres.
the centrosomes or the centriole pair.
Oh.
What do they do?
They help with the
Protein synthesis.
No.
and the microtubules and
But think of cell division. What's their job in cell?
metabolism.
Not
No.
metabolism. In cell division.
Dang it.
The spindle fibers.
Spindle
Fibers,
yeah.
which the chromosomes move along. All right, number 3. We didn't mention number 3, so I'm just going to tell you what it is. It's a vacuole. That's one of those sort of extra structures. And the vacuoles are storage for the cell, or will supply storage for certain structure or materials, and they also help with waste disposal. So what's 4? 4 is just indicating all this stuff.
Cytoplasm.
It's the cytoplasm, okay? 5. 5 is a rounded structure,
Nucleus.
Nucleolus.
nucleolus. What's in the
Right.
nucleolus?
The nucleus.
The
No, the
What?
nucleolus is in the nucleus, but what's in the nucleolus?
mRNA.
mRNA. Okay. So if 5 is the nucleolus, what's 6?
The
6 is
nucleus.
the
The
envelope.
nucleus itself, or the way it's pointing, you could say it's the nuclear membrane or the nuclear envelope. Either of those would be fine. All right, 7. It's just sort of a little rounded thing.
7
Lysosome.
is
Either a lysosome or a
The
prednisone.
lysosome. It's the lysosome. So what is the lysosome? What do they do?
they take out the garbage.
Exactly. The garbage disposal. All right. 8. They're on the outside of the cell membrane, and
Yeah.
they
Looks
allow
like a
movement.
right.
The cilia.
Oh, the
Cilia.
nuclei apparatus.
The
No,
cilia.
the
Nuclei
cilia.
They
apparatus.
say silly.
No, that's
Cilia,
not it.
No, 8 is
Oh, that's. Oh, okay. I see what you're saying.
all right. 9 is indicating this whole
Ribosomes.
Right.
nope. This whole structure with the little dots on the outside.
The
Smooth.
rough.
ER.
Endoplasmic.
The rough
ER, the rough
endoplasmic
part.
reticulum.
Smooth
Okay.
reticulum.
I'm sorry.
Yeah.
Okay. All right. So that's 9. This whole thing's the rough endoplasmic reticulum. So what's 10? That are these stacks of flattened
Smooth.
stacks.
Googly eye apparatus.
Not googly eyed.
Yeah.
Smoothie R.
Golgi.
It's not.
These are the Golgi.
Golgi. Oh.
10 is the
Yeah.
Golgi apparatus, the Golgi bodies.
Well, that's what I meant. Same thing.
That's what you meant. You just couldn't get the words right. 11.
11. Where's that at?
These kind of straight line looking things.
Microtubules.
Microtubules. Okay, 12. 12 we didn't talk about and you can't see them clearly. It's something called the Golgi vesicles. Don't worry about them because I can't even differentiate them. So what's 13 if it's these little dots?
Ribosomes.
Ribosomes. All right, and then what's 14?
The smooth
A
ER.
little dot.
Smooth endoplasmic reticulum. Okay. All right. Let me see. I'm going to ask you some questions. We're just going to go through, just do a little bit of review. Some we've already answered, so I'll have to pick some. All right, first one. Where does protein synthesis occur?
The
Ribosomes.
On the ribosomes, okay? For the cell biologist, what are the 4 stages of the cell cycle?
Metaphase. I mean prophase, metaphase, anaphase.
Nope,
telophase.
that's cell division.
M
Fourth.
phase G1 S G2.
Oh,
All right,
that,
M,
yeah.
G1, S, G2. What's another term for carbohydrates?
Glucose.
That's a term.
Sugar.
They are sugars, but
Macromolecule.
formal term.
Macromolecule.
The saccharides.
Oh,
Carbohydrates are the saccharides. Remember monosaccharides, polysaccharides?
Disaccharide.
What are the 4 major organic compounds?
carbohydrate.
Lipids.
Water.
Nucleic acid.
Lipids,
Water isn't one
Protein.
because it
Water's
don't
not
contain
one.
carbon.
Carbohydrates, proteins, nucleic acid.
all right, so lipids, carbohydrates, proteins, nucleic acids. Which type of cell division is termed reductive division?
Meiosis.
Meiosis.
because you start out with twice the number of chromosomes. They're cut in half, and they're cut in half again. So you're reducing
The
the number of
haploid.
chromosomes. What is a more common term for lipids?
Fat.
Fats.
Fats. Okay. $22 different what are involved in protein synthesis?
Chromosome.
Amino acids. Proteins are made of amino acids. All right. Let's see. Approximately 80 to 85% of the human body is what?
Water.
Water. Okay.
Water.
What occurs in S phase of the cell cycle?
DNA replication.
DNA replication.
Yep.
What part of the DNA molecule is made up of nitrogenous bases?
The rungs.
The rungs or the steps. All right. Let's see.
Can you repeat that question?
I said, what part of the DNA molecule is made up of nitrogenous bases? And it's the rungs. Which of the macromolecules provides fuel for cell metabolism?
Carbohydrate.
The carbohydrates, exactly. Proteins can act as what three things, or what? There's three. Proteins are different kinds of molecules, but what are the three main ones?
Is this the energy one?
That was poorly worded. I can't quite decide how to say how to ask the question of how I want it.
Lipids.
But protein molecules function as what three things?
Enzymes.
Antibodies, hormones, and enzymes.
Exactly. Enzymes, hormones, and antibodies. What's the purpose of a hormone?
Homeostasis.
Well, they're all,
Balance.
well, that's too broad. Give me more specific.
Regulation.
Regulation. That's right. They're regulating body function. What are the chromosomes composed of?
Amino acids.
DNA.
Oh, I was right. Oh, I guess I was laughing.
Hey, you got it. Good job. Let's see. What's considered the control center of the cell?
Mitochondria.
Nope.
No,
Nucleus.
nucleus.
The nucleus. The nucleus is the control center.
How many hours does it take for reproductive cell division? 15.
How many hours? Yes. How long does it take for cell division? All right, Marcus, you asked the question. How long?
15.
1 hour. How long does it take for DNA replication?
15 hours.
15 hours. That's right.
DNA.
Okay. Well, do y'all have
Nope.
Yep.
questions?